Morning. Glad you guys figured out that it was still at 8 a.m. even though I made a mistake on the Zoom. But I think I set all the right numbers. I just set up the Zoom for 11 instead of 8. Wishful thinking. No. 8 a.m. Here we are. And it's our usual suspects. Love to see you all, ladies. We're going to jump right in because I want to try to squeeze 2 short FRQs in this morning. Just because I feel like they're giving the same attention to both Unit 5 and Unit 6 as the rest of the units as far as the review goes for the AP, which of course that makes sense because you're supposed to study everything evenly. But I personally believe that Unit 5 and Unit 6 are 2 of your harder units as far as concepts go and content. There's just a lot going on in both of those, especially with the genetics, like DNA synthesis and stuff like that. So anyway, there's a lot of FRQ options out there. I think the more we see, the better chances we're going to have. So I'm just trying to squeeze in some extra ones here for Unit 5. So with that said, we will jump right in. Not much to say about the first one. I will just pop it up on the screen and chat about it and read through it with you, and then I'll turn you loose. It's a describe. It's our favorite. It's storytelling, lots of details. But now I have to find it. Stay with me. Okay. Great. So here's our first one. Again, it's a short FRQ, so you have 15 minutes. It says, let's see what it says here. Come on, get out of the way, you silly thing. There we go. Both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from the parent cell. So far, things we know. A, describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. So let's just talk about that one really quick. Again, we're going to describe, so we're storytelling, lots of detail, lots of concise verbiage, painting a picture for our reader. If your words aren't great at painting a picture and you're writing, you know what kind of feedback you get from teachers about your writing. If you know that you're not great at painting that picture with your words, then you should draw a picture. And again, as far as science drawing goes, there's nothing too artistic involved. Circles and lines can represent chromosomes, no problem in a cell. So draw a picture in the case of these if you're worried about your words. 2 events, don't do any more, don't do any less, do exactly what they tell you to do. 2 events. And then what are we drawing events about? It's not, we can easily jump to conclusions here and be like, oh, draw 2 things that are different between mitosis and meiosis. No. Read it very carefully. They want to know events that happen within both of those. So both of them have to have the same event that ensures the resulting daughter cells inherit the appropriate number of chromosomes. So how do we know that we're going to do cell division, whether it's mitosis or meiosis, and get the right chromosomes in the daughter cells? And there's lots and lots and lots of different ways that the cell organizes this and makes sure of it, but you only have to talk about 2. Okay, B says the genetic composition of daughter cells produced by mitosis differs from that of the daughter cells produced by meiosis. Okay, no, so now we're doing, comparing and contrasting these 2. Describe 2, no more nor less, 2 features of the cell division process that lead to these differences. Oh, great, now I can highlight the differences between meiosis and mitosis, right? So when basically it's saying at the end, the composition of the cells is going to be different between mitosis and meiosis. We know that. That's the reason there are 2, is because the 2 different styles of cell division are going to come up with 2 different products at the end. describe how that happens. How do we start with the same but end with different? Alrighty. So again, this is a describe and you have 2 events for each. I'm going to say this is probably minimum an 8-sentence FRQ, right? But at the same time, it's a short one, so you only have 15 minutes technically. So I'll turn you loose, I'll shut up, start writing, start jotting things down, start drawing yourself some pictures. Remember, if you can write more now and then delete and edit and get some feedback on it, then when it comes to the real thing, you're going to have the exact answer you want. So it's okay to stumble through, and it can be ugly and messy today. Ooh. I want to navigate to another screen, but I want to make sure I don't lose this for you guys. If I change my screen, what does that do to you? Did that change things, Sadie? Oh, okay. Never mind. That's why I have 2 computers. How we doing? Just checking in. Just wave a hand or something if you need more time. Yeah, absolutely.
I need more time.
Yeah, definitely. It's only been about 8 minutes, so totally fine. You can have another 8 if you need it.
May I see the second question again, please?
Oh yeah, I'm so sorry.
Thank you.
You got it. There you go. How we doing? Anyone need more time?
Yeah, like 2 more minutes.
Totally. Yep. Take your time.
I'm done.
Cool. Michaela, Moira, how you doing?
I just need like 1 more minute.
Take your time. Totally. Moira, just checking in. How you doing? Thumbs up. Great. Okay, we'll just start chatting about this one. And then if you have some questions, we can go over them. The first part, I'm just going to put it back up. I don't know why. You need to see my face for any reason. Okay, the first part asks us to describe 2 events that are common to each other between mitosis and meiosis that ensure the appropriate number of chromosomes in the daughter cells. Anyone want to talk about one of the events? You don't have to tell me your whole complete sentences, but just maybe what one of the events was that you described.
I said the aligning of chromosomes during metaphase.
Beautiful. Yeah, metaphase lining up. That's great. And that happens in both mitosis and in meiosis in both the first and the second phases. So great. Anyone else?
I'm not really.
I said anaphase. Oh, sorry.
Go ahead.
I said anaphase and making sure how that half of the chromosomes are like the sister chromosomes are pulled to the opposite sides of the cell.
Perfect. Great. So you talked about the disjunction that happens in anaphase. Good. Sadie?
I said, I'm not really sure if it counts, but I said the duplication of chromosomes before both processes.
Yeah, absolutely. So just the fact that we have DNA replication or synthesis before either one of these starts, the actual separation into 2 new cells. Yep. Is that what you mean? Did I get that? Did
Yeah.
I? Okay, great. Yep, good. Anybody else? Something different? Okay, I'm just going to name a couple more just in case, so you can just jot them down in case you're like, oh man, yeah, that's much easier to talk about or easier to draw a picture of, whatever you want. Those three that you mentioned, though, were great. So far we already mentioned lining up during metaphase. That's just kind of like the check, right? Let everybody line up in the middle so that we can separate accordingly. Good. We talked about disjunction, which you could have gone into, maybe some more detail about, the cell contains spindles and a microtubule that reaches out and grabs the specific sister chromatid at the centromere and separates it. Whatever, you could draw some pictures of that. But disjunction in general, that separating of the two sisters, depending on what phase you're in. You can go into detail about the different cellular structures that are involved in that. Let's see, we talked about just pre-mitosis and meiosis, which is borderline a little, just on the edge of what they're asking, because they do ask about events that are common within mitosis and meiosis, and technically that condense the DNA replication and the condensing of all the chromatin could be seen as right before both of those phases. It's that gray period of, is interphase part of mitosis or not? Is it on the outside or is it in the inside? No, depends on what field. I think you'd be totally fine talking about that. But just saying it's right on the edge. Let's see. In general, we kind of, when we talked about disjunction, sort of, you could talk about the telophase where cytokinesis takes place and separates it into two separate cells. Let's see, you could talk about, again, not really in mitosis and meiosis, but again, right on the edge, on the outside, we start that G1 checkpoint just to make sure that it all happened correctly. So again, not within the actual phases, but right on the outside is the check to make sure that those phases happen. So you might have talked about G1 checkpoint. That's it. Those are the ones I got. Those are the ones you got. I think those all sound fantastic. Any questions about A? Great. Just wondering, did anyone draw a picture? Okay, just wondering. Okay. B is now we get to compare the two, or contrast them, really. We want to look at two features of the cell division process within mitosis and meiosis that make them different in the end. Obviously, we know we go from, having the same number to the same number, ooh, magic, splitting a cell but somehow ending up with the same number in the end in the two new daughter cells, to essentially cutting the number in half and ending up with 4 new daughter cells, okay? So how does this occur? Does someone want to talk about, we don't have to read your exact sentences, but maybe one idea or concept that they mentioned?
I said crossing over / genetic recombination.
Great, so which one can do crossing over?
Oh yeah, I talked about how meiosis does
Great.
crossing over.
Perfect, great. So we have crossing over that it can occur in meiosis. Don't mind my street sweeper. He's getting really good at dodging my car. Almost too good, like just a little too close. But that's fine. Good job, buddy. Great, thank you, Jayla. So crossing over that happens in meiosis. Somebody else a different one?
I put that in metaphase of mitosis, sister chromatids line up, but in metaphase 1 of meiosis, it's homologous chromosomes.
Yeah, exactly. So the lining up and the different metaphases. Oh, and you drew a picture. That's exactly right. Look at how beautiful it is.
I don't know if you can see it very well.
Oh, I totally can.
Okay.
Yeah, 100%. Yep. So basically she's got in her mitosis metaphase, she's got her double wide sister chromatids, and then in her metaphase meiosis or meiosis metaphase 1, she has her homologous chromosomes, so quadruple wide, paired up and lined up so that they can do that type of separation. So perfect, that's great. Just the two, like if you just used one of your features as just comparing, or I should say contrasting the two types of metaphase, that's fantastic. And a picture to boot, even better, even more clear for your reader. Great, so we've got crossing over, we've got chromatid separation.
Miss Mason, I have a question.
Yes, please.
So you know how yesterday they did the test, like practice uploading?
Yes.
So, oh shoot. So if we if we do like the one on the document, how would we insert pictures?
If we do the one on the document,
Like where you do it on Google Docs and then you upload it,
yep.
instead of like writing it on paper and taking a picture.
Yes.
Like how would we draw a picture if we need to do one?
Gotcha. So, okay. If you want to draw a picture, but you're using Google Docs, my best suggestion to you is to draw your picture on a scratch piece of paper, take a picture, and upload it to your Google Doc.
Oh, okay.
Does that make sense?
Would they let you upload like to Google Docs? Okay.
Correct, yeah. You could also, if you draw the picture on the paper, take the picture, put it on your computer, upload it to your computer, then you can like drop the picture into the same doc that you were using in Google Docs.
Oh, yeah, okay.
Yeah, but this is where like Notability is going to be gold, because you can type in Notability, you can format in Notability, you can drop pictures into Notability, you can draw your own pictures in Notability, you can graph in Notability. I mean, like, it's going to be a good one. You can change colors. I think that's going to be your very best bet, because then you're going to be able to have that PDF and just have it right in. And if you're more comfortable in Google Drive, especially since you can type more freely, that's totally fine. I mean, especially like if you want to use your computer versus using your iPad.
Yeah.
Okay. And I totally get that. But you could also have your iPad to draw pictures and then, throw them over to your computer.
Thank you.
Yeah, and we'll practice that too a bunch next week, like just the timing of it. Because that's the part that can be a little bit more stressful. If you take 15 minutes to answer your question and now you are only left with 5 to upload or whatever it may be, then, like if you're frantically trying to pass this picture to this file and then download that file and then something glitches, like you don't want to be stressed about that. So we'll practice that a bunch next week. Okie dokie. Anyone have any more ideas about how these two are different? Features of cell division process. So far we said chromatid separation during metaphase, and we talked about crossing over that happens only in meiosis. One of the more obvious ones, that maybe you overlooked because you think like, well, duh, they know that, but it's just the fact that in meiosis, they go through 2 cell divisions instead of just 1, right? Mitosis is we're just gonna split in half, done. Whereas in meiosis, we have, okay, we're gonna split in half, and now those 2, we're gonna split those in half again. So basically 2 cell divisions versus just 1. Doesn't seem like it, you're like, duh, that's obvious, but jot that down and you nailed it. Draw a picture, whatever you need to do. Questions about part B or anything else to add to part B? Okay, I'm gonna throw up our second one, even though we only have like 5 or 10 minutes left. We won't get through it by any means, but at least you can see it, you can take a screenshot of it, you can we can just go over it really quick. Yeah, I'll just shut up and we'll just do that. Let's see. So this should be Unit 5, practice number 6. You know what? Well, okay, while you're looking at it, I will also send it to you just because it is a little bit wordy. You know what? No, I won't send it to you because I did my due diligence yesterday, you guys, and I uploaded all of the FRQs in the world to our haiku. That's not true. I'll still send it to you anyway. Okay, so let's just read through this together. It says fruit flies, Drosophila, melanogaster. I love the name of fruit flies, Drosophila melanogaster. They sound so cool, but it's just a fruit fly. But anyway, okay. With a wild-type phenotype that have gray bodies and red eyes. Certain mutations can cause these changes to these traits. Mutant flies have black bodies and/or a cinnabar eye. To study the genetics of these traits, a researcher crossed a true breeding wild-type male fly with gray body and red eyes, so we're going to go total dominant, homozygous dominant on both, with a true breeding female fly with a black body and cinnabar eyes. So that just right off the bat, that might not be super clear to us because we're used to saying things like homozygous dominant, homozygous recessive, or heterozygous, right? When researchers start talking about true breeds, it means that they're fully dominant. They have that trait completely. So when it says true breeding wild type male, gray body, red eyes, they're going to be capital G, capital R, capital R. Okay? And a true breeding female fly with black body and cinnabar eyes, which we're assuming are the mutants, right? You would consider that to be lowercase g, lowercase r. Okay, I just assigned those letters arbitrarily based on the phenotype, but I just want to make that clear. Okay, so then they crossed those two, all F1 progeny displayed a wild type phenotype. So you probably did that Punnett square in your head and you're like, yeah, that makes sense. I'm going to have true heterozygous all across the board because I did 2 full homozygous parents. Okay, female flies from the F1 generation were crossed with true breeding male flies with black bodies and cinnabar eyes. Okay, so that's, again, lowercase g, lowercase r. So we have a heterozygous crossed with our recessive homozygous. The table on the next page, or right here, represents the predicted outcome of the data obtained from the cross. Explain the difference between the expected data and the actual numbers observed. So this one might seem a little tricky at first because you're like, oh my gosh, all these crosses, I gotta do a Punnett square, look at all the different outcomes there are, the prediction doesn't match the observed, I must have to prove it with a Punnett square. So as you're reading through this, sure, would it be a good idea to maybe do a Punnett square for these right now while we're practicing? Yes, great opportunity. But then you're gonna come to find that later on, that's not what they're asking at all. At the very end, after we read through all of that, all they want you to do is explain why the prediction is going to be different from the actual observed when it comes to genetics. This is really just a test of what you know about genetic outcome. It isn't necessarily about doing the math for the Punnett square. It's just knowing, okay, if that's what I was supposed to get, if I was supposed to get 25% of each of the different crosses, or each of the different, breeds, then why did I only get, why did I get a 50-50, 1-1 or whatever this weird ratio is, right? Why would I not get more of that? Does anyone want to just speculate without necessarily going into great detail or writing it down right now?
Is it because they're linked or something like that?
Yeah, linked or something like that is a great answer. I mean, you're going to say it much better, right? But yeah, exactly. There's a good chance that these two genes are so closely together on the chromosome that they like to stay together when they're being passed on. And so that's why we end up with, the red and the gray staying together and the black and the cinnabar staying together. Sadie? Were you going to say something? Sorry.
I was going to say the same thing.
Same thing, yeah. Another thing you could just talk about in general is the fact that each individual selection for an offspring is independent, right? Every time the offspring happen, it's a 1 in 4 chance of what you're going to get. So it's not necessary, genetics are a great way to say the probability of what's going to happen, but it's not always what's going to happen, right? So you could talk about both of those things, but you nailed it. So this is just a good example of a question where It seems like I have to do a Punnett square and show math, but then you're going to waste a ton of time doing a Punnett square just to say what you said at the end, which is, oh, these genes must be linked, or it just so happened that in this particular cross, this was the outcome, because each individual selection for a gamete is independent. Okay, I'm going to send you this one so that if you want to write yourself an answer, you can. Otherwise, we are done if there are no more questions. Questions?
I have a quick question.
Sure.
Is Unit 6 the last unit that we have to know for the test?
Yeah. The only thing that could potentially pop up from 7 or 8, I guess it would be 8. The only thing that could pop up from 8 that they mentioned in some of our webinars was like statistical data and looking at graphs, which we're doing a lot of anyway, and you guys have gotten pretty good at that. And it's not something that was specifically covered in Unit 8, but like you kind of did more of that data analysis because we were looking at so many evolutionary trends. So like, you know, the fact that we did the lizard biointeractive was a good one because you had to look at a ton of graphs and stuff like that. So you guys have those skills down, I think. And when we've looked at graphs, I haven't seen too many issues with that, but that's the only kind of concepts that they might pull from 8. Nothing about evolution, nothing about, you know, ecosystems or communities or anything like that, but just the idea of looking at statistical data. Cool. Anybody else? Okay, I will send the 4 of you this direct FRQ, and then you can send me back if you want feedback.
Can we do you know that there are FRQs on like the College Board website?
Yes, College Board has FRQ practice.
Can we do FRQs from there?
Oh, sure.
Like, not with the class, but like, you know, like in the morning sessions.
Sure,
Or
I can pull some of those instead. All of the ones that we have been doing are College Board FRQs as well. They're just, who knows when they've been pulled from. I don't know what, like I've just been pulling them from the live sessions, the live College Board sessions. So, but yeah, I can pull some different ones from past years for sure. Cool. All right, I will see some of you in just a little over an hour in class. Alrighty. All right, see you in a bit.
Happy Teacher Appreciation Week.
Oh, you're so nice. Thank you.
You're welcome.
All right, I'll see you in a bit.